Hello, and welcome to The Issue on the ASGCT Podcast Network. I'm your host, Emily Walsh Martin, and today we're tackling my absolute favorite subject, the importance of long-term data. Dr. Dolan Sondhi of Weill Cornell Medical College joins me to discuss her recent paper, 20-year survival analysis of Adeno-Associated virus vector serotype 2 mediated gene therapy to the central nervous system for CLN2 disease. I'm so excited to have this conversation with Dolan because long-term follow-up is so under-discussed and under-available in our field. I'm thrilled to share my conversation with Dolan and encourage researchers everywhere to think creatively about how to collect data for as long as possible from your clinical trials, even if it is just an annual phone call. And then, of course, ultimately publish your long-term data. And while we're at it, I hope to see all of your long-term data at the ASGCT annual meeting, the premier event in cell and gene therapy. The Society is headed to New Orleans this May, the 13th through the 17th, with more than 2,100 abstracts submitted for presentation. You can save $465 by joining ASGCT and using the incredible discounts the Society provides to members. Check out the full program, plan your trip, and register for the ASGCT annual meeting by visiting annualmeeting.asgct.org. And now on to our episode. Hello, and welcome to the podcast, Dolan Sondhi from Weill Cornell Medical College. Thanks so much for joining to talk about literally my most favorite topic in the world, which is publishing long-term data from gene therapy clinical trials. But before we dive into your long-term data, can you share your career path a bit with our audience?
Yeah, absolutely. Thanks so much for having me on, Emily. And well, I've been in the gene therapy field for 25 years, and so also long-term data right there. But I sort of ended here a bit not your normal path. I'm actually a PhD chemist, and I slowly made my way into more biological arena and ended up at Weill Cornell, where I was hired as an assistant professor in 1999. And, very soon after starting here, so it was the Department of Genetic Medicine. And very soon after starting, we were at I think it was actually an ASGCT meeting. If not, it was an NIH meeting. It was one of those two meetings. I don't fully recall. And we were, I was with a group of faculty members from my department and the chair of my department, Ron Crystal. And we were approached by Paris Bird from the FDA. And he said to us, would you guys be interested in pursuing neurological gene therapy for a rare neurodegenerative disorder? Until then, we had really not done any CNS gene therapy at all. And we sort of looked at each other and said, maybe let's sort of look into it. And since I was the new kid on the block, had the most time, didn't have any like long-term projects yet, I was given the baton and said, fly with it. And I guess 25 years later, I've done a lot of CNS gene therapy. And that was really the start of it. And I can tell you more about it as we talk about the paper. But it has been, I would say, the most exciting 25 years because I'm hugely passionate about gene therapy, especially adeno-associated virus-mediated gene therapy, and especially as it relates to the brain.
Absolutely. And when you say, you sort of dived into it, like you've sort of seen all parts of this. So you have seen the CMC parts, the non-clinical parts. Talk a little bit about how cross-functional you've been in your career.
Yeah, so that actually is so much a function of our department. And so the way the Department of Genetic Medicine is set up is we have our department and we have also a core facility, which is the Belfer Gene Therapy Core Facility. And between these two entities, we have essentially the entire infrastructure to be able to take an idea from an idea all the way to the clinic. And so that really involves, we'll say, okay, which disease do we want to approach? Why do we want to approach that disease? We create the molecular biology constructs, we make the viral vectors, we then do all of the proof of concept efficacy studies. We have an animal facility that is dedicated just to our work. So nobody else uses that facility. So it's fantastic. We have a lot of control over it. And then onward to thinking about, okay, this data is starting to look good. So having those initial regulatory discussions with the FDA. So I've been involved with all of those through pre-INDs and such. And then going on to safety and efficacy studies. And so what we have, which is very unique, is also being able to do all large animal studies in-house as well. So we can do non-human primates, we can do pigs, we can do you name it. And so we have the ability to do that. And any kind of imaging modalities that may be required, we have facilities to do that. So that happens in-house. So we've done our safety toxicology studies. We sort of do it GLP. So I've got to really see all aspects of that and actually lead most of those studies myself, especially all of the animal work. And then this is the most fantastic part of the whole thing is we have a GMP manufacturing facility. So we are able to make our drug product. And this everybody finds it surprising because we have a GMP facility on the 10th floor of a Manhattan building, but that's how you have it. And
Wow.
with 3 manufacturing suites, so we are able to make product, and this product has been we've made it for our clinical trials and for others. So 20 times, I think we've been in the clinic so far with product that has been made in our facility. And then, so all of the INDs and everything. So I've been involved putting that in, having all the conversations with the FDA, then helping our chair, Ron Crystal, who is, the sponsor of our clinical studies and an incredible mentor. We sort of all work together and we put together a clinical team that will lead the sort of project. So then we run the phase 1 studies, have had the opportunity to further license these things out to biotech. So really, as you said, Emily, we have the infrastructure to do it from the whole way through.
Yeah, no, that's amazing and somewhat a little bit rare because oftentimes, if you have a role, let's say on the private company or public company side of our industry, sometimes the product has already been ideated somewhere else and shows up, and then you're diving in. But you've really gotten to see things from sort of idea all the way through, which is pretty spectacular.
Yeah, very exciting. It's a great place to be.
Yeah. And not only that, because of your longevity, at Weill Cornell, you've also been able to see things sort of progress for a long time. And that sort of dives us into your recent paper, which is entitled 20-Year Survival Analysis of Adeno-Associated Virus Vector Serotype 2 Mediated Gene Therapy to the Central Nervous System for CLN2 Disease. And you're the first author for the paper. I want our listeners to appreciate that this is probably either the longest term follow-up data that's been published or some of the longest data that's been published for AAV2 vectors. And in this case, you're using it to treat Batten disease. And so can you talk a little bit about CLN2 Batten disease, maybe to get us started, the genetics, epidemiology, what these patients sort of how they present and what they go through?
Yeah. So CLN2 disease, well, we used to call it Batten disease back in the day, but it's, the new terminology for it is CLN2 disease. And so it is caused by mutations in the CLN2 gene, and this leads to the deficiency of the protein tripeptidyl peptidase 1. And the absence of this is extremely harmful to neurons more than anything else. So it's a lysosomal enzyme, and the absence of it leads to waste products not being degraded and collecting in the lysosomes and leading to neuronal cell death and everything sort of that follows. And it's really sad because when these kids are born, they appear normal, but around age 2 or so, they start seeing a lot of regression in gained knowledge as well for these kids. They sort of start losing language, they become have motor issues, they become wheelchair bound, they also go blind, and oftentimes they die between the ages of 8 and 12. It is an autosomal recessive disease, so both parents need to have the mutation for the child to have it. And I especially mention this because this is also extremely devastating for the parents because they have seen their child to be normal to start, and then it really goes downhill. And a lot of them, because both the parents have the mutation, they also feel somewhat responsible and oftentimes become very keen to be able to do something. And so there is a lot of involvement of patient foundations in progressing therapies for diseases like this. And we had a huge involvement from one such foundation called Nathan's Battle Foundation, and that's how this project got started almost 23 years ago, actually.
Yeah, absolutely. This is one of a number of diseases in this family of CLN2. There's other CLN
Yeah.
disorders as well, and they all have slightly different sort of timing of onset, but the sort of conceptually very similar in that, the children are doing okay, but then they start to lose skills, regress, have seizures, etc. And obviously, it's sort of every parent's worst nightmare, and they want to be able to do something. So in this study, you talk about you've treated 10 patients, and they were treated with an AAV2 construct containing the affected gene. And in this case, you're actually delivering the AAV2 gene therapy directly to the CNS. And can you talk a little bit about the route of administration? Because it's somewhat unique compared to other AAV therapies.
Yeah, so I think we need to go back to 2003 when so this project sort of began in about 2000, and we moved very quickly through the paces to get all aspects of it done. Fast forward, we are at 2003, we have all aspects done, and we submit our IND to the FDA and in December 2003, and we get allowed to do this in January of 2004. But with the knowledge that AAV2 was really the only available serotype, in addition to other first generation serotypes like 5 and such, there was no AAV9. We had not yet gotten to the stage of thinking about vectors that are able to cross the blood-brain barrier. We had also not started thinking about delivery to the CSF. So this is sort of very, very early on. And the idea was if the gene, like if the major culprit or the major organ where the disease is the brain, the real best way to access the brain is to deliver to the brain parenchyma. And so the two organs of interest to us when we first started were the brain and the eye, because the children also go blind. And there was some initial discussions about doing, gene therapy to to either or both. And the final decision was to proceed with brain gene therapy to the CNS, to the brain, because that was what was causing essentially the early death and the main sort of phenotype. And so the best way to access the brain was to deliver to the brain parenchyma. And then we started thinking about the fact that how do we access as much of the brain as possible, because the disease is widespread. And so the idea was, and this came through with discussions we held at this time. We had so many KOLs actually visit us at our department, and we had them give talks about the disease and learn more about it and see what would be the best way to approach this. And was the idea was, okay, we need to deliver in as many regions of the brain as we can. And then we met with neurosurgeons and decided, okay, what would be really the best way to do this? And so we decided to proceed with 6 burr holes, and to maximize distribution through each burr hole, we delivered at 2 depths. So essentially, you can think about the fact that we were delivering at 12 locations. Wow. A very important thing to remember, though, is that TPP1 is a secreted protein, which is fantastic, because you have the bystander or the neighboring effect. And essentially, this is the mannose-6 phosphate receptor pathway. So TPP1 gets secreted from the cells that are infected by the viral vector, and then it gets picked up by neighboring cells, and those cells also get corrected. So if you were thinking, if you had, say, a dime size region of actual infection, you may get a quarter size of correction because of the bystander effect. This is just, these are not real numbers. I'm just giving an idea of what it would be. So you would get increased, essentially, distribution. So we took all of those things into account as we decided how to do it. And then it was all put together with the expertise of 2 really fantastic neurosurgeons at New York Presbyterian Hospital, who were supremely involved in this process. And essentially what it involved was, obviously, we had to do a lot of mock runs, and you don't even want to know how we actually had to figure out things that would be sort of reminiscent of a human brain, and kind of set up mock situations where we would practice this first with the surgeons. And essentially, what happens is when the patient would come in, they would get MRI the day before, within 24 hours, essentially. And that would help plan the trajectory of where we would be inserting the catheters to deliver it. So essentially, our setup would be, it was a pump with 6 syringes that have catheters coming out, flexible catheters, that then are put through flexible arms in a head frame on the child's head, and through a spinal needle, then this catheter goes through, and that is then delivered at 2 depths, at a lower depth, then pulled out, and then delivered at a less depth, a deep area. And that's how it was done to be able to access 12 locations. But this was different for each child because of the neurodegeneration. Their parenchyma was different.
Got it. And so because each of these patients may have already had some level of neuronal loss, their geography is different. And so each case had to be planned specifically for that patient, is what you're saying.
Exactly.
Wow. And were these all administered at the same site? Was that?
It was a single site study.
And same clinical site, obviously 12 sites, but
yes, same clinical site, a single site study, same neurosurgeon performed all all of the surgeries, and same neurosurgeon planned all of the studies. So there were two neurosurgeons. One did all of the planning, one did all of the administering.
Okay. And, obviously today it's maybe a little different, these sort of assumptions about immune suppression before providing an AAV. In the case of this therapy, did you guys do any pre or post immune suppression or not at all?
No, no immune suppression, because
Okay.
at this time, two things. One, we all started with this thought that both the brain and if we had done the eye are immunoprivileged organs, and that we would likely not have an immune response. And when we started this about 22 years ago, 3 years ago, there were really, we hadn't come across all of these later immune issues that we have, come up with. And in follow-ups of these patients, we really did not observe any systemic immune issues. We have previously talked about in various different talks, as well as there are a few other published papers about this kind of setup and this treatment, where we have observed local T2 flares by MRI where we administered, but there were no clinical sequelae to these.
Okay. Yeah. And so maybe now let's dive into the results of the study. So as I mentioned, there were 10 patients who were treated, and what was interesting to me about your new paper is that the response to the therapy seemed to be divided. There were kind of two groups. Tell us a little bit about that.
Yeah, so everything should be taken with a grain of salt, because we obviously made this discovery post treatment. We did not plan it. We did not plan it like this. But of our treated subjects, we did something different, in that even though the mandated follow-up is currently it's 5 years post treatment. So our clinical trial ran for 18 months. We had 18-month follow-up with all kinds of tests, and them having to come back and do MRIs and all of the different kind of neuroscales and such. But then we followed up with subjects annually and then periodically, just to see how the kids were doing. And when it was last summer, we found out about the last person in this trial, when this child passed away. We kind of put all of the information from all of the patients together, and in looking at it, observed that 3 out of these 10 children lived significantly longer than not just the control subjects, but also the other treated children. And so it was 3 and 7 is how it happened. And 3 of the children, so the normal range of kind of the age by these untreated children passed away was between 8 and 12. And many of our treated children also, sort of passed away around the same time, even though they did see improvement in the neuro rating scale. But 3 of the patients lived significantly longer, in that they lived to the age of between, I am actually looking at the paper because I don't remember top of my head, but about 18 years to 27 years of age. So a lot longer than the rest of the group. And so that really kind of sparked our interest as to what was different about these kids. Why is it that they lived longer? Was there anything different between their kind of genetic makeup as far as the disease was concerned, or their disease status when we first enrolled them in the study? So we looked at various things, and this was obviously done after the fact. And so, this isn't a paper where all of the information was collected as planned. I mean, all of the information was collected as planned, but this is all information that was collected after that time, that I'm talking about in this paper. So,
Right.
and so that's what we started thinking about, and that's how this paper came to be.
Yeah, absolutely. And so, yes, I mean, I think you're right to say, and it's very good of you to say. Obviously, this is a post hoc analysis, but at the same time, right? This is a disease where there is a great deal of natural history, and where I think even though this wasn't an intense level of follow up that you and your team did, it was sort of an annual check in with the patients and their families. It's still such important data that you've discovered where, again, you're falling into two groups, one group with seven patients who basically had, I'd say, similar response to being, either treated or untreated. Those patients sort of had the same survival trajectory as untreated patients, but then the three patients who actually turned out they were the first three patients dosed, right?
Yeah, yeah.
And one of the things that I noticed, and I think you guys also note in this, is they happen to be a little bit older, and the most interesting part though was that they happened to potentially be, while they had very similar, let's say, disease manifestations as the other seven, they did have a difference in ventricular volume, or which is the inverse of brain volume, if you will.
Yeah, yeah,
Yeah, neuronal volume.
yeah.
So talk a little bit about some of the hypotheses that you guys looked at, and where do you think this data is pointing you in terms of what might explain the difference between these two groups?
Yeah, so it was really, interesting because there are the first thing that we thought about was really the genotype, right? We have a paper that we published with a group in Hamburg, which is in Lancet Pediatric Neurology, and we have, I think, together between our group and their group, over 100 patients that we have natural history data on in that paper. And we really hadn't been able to establish a real genotype phenotype correlation. We didn't see that. And so, but we do know that there are two mutations that really are predominantly present in about 60% of the children with this disease. And so we said, okay, let's look what's going on here. And these three patients did have the same, one of those same, the most common genotypes, and their second genotype was, the genotype on their second allele was different. And so then say in the other seven, who predominantly had those two major, one or the other of those two mutations. But then we looked at their combined annotation dependent depletion score, CAD, which is something that is often used right these days to see how deleterious is this mutation likely to be. And essentially their scores matched. Both the groups had pretty much the same score. But we said, okay, well, let's also look at another control group. And this control group was like our three long surviving subjects, in that they had only one of those common mutations, and their second mutation was also different. And they also had the same kind of deleterious CAD scores. So, but they had what was like the regular survival for these patient population. So essentially, gene therapy, we do know, made an impact, right? Because there was something going on there. So genotype alone was not the cause of this difference in survival that was observed. So then we were like, okay, so what other things? Their starting scores were on the neuro rating scale were similar, so that there were a lot of similarities in these patients. But what was, as you said, different was we did observe that these children were older. They actually had greater neurodegeneration because their ventricular volume was higher. And so this made us start thinking about our more recent data where we have administered the same for CLN2. We have a paper that we've published in non-human primates where we've shown that if in comparison to delivery via the intraparenchymal route, if we deliver via the intracisternal route, so if we access the CSF or the ventricles, essentially the CSF route, that we get a lot greater distribution than when we were doing our parenchymal route. And so a question that came to mind, is it possible that in fact the reason for this greater survival is that we are getting greater distribution, which, could be, but we don't have a way of proving this. And it actually goes against my other paper that we sort of published a while ago, where we showed that the earlier you treat, the better outcome there is, right? So we have another paper where we treated disease knockouts mice as neonates at 3 weeks and at 7 weeks, and the neonates did the best. So that's kind of different than now the observation that we've made right here. So that's what made us think that perhaps it is really something to do with the biodistribution and all of that. And it would have been great, Emily, if we had been able to get perhaps some tissue or something to be able to analyze that. And we had, and I think we talk about this in the paper, these are some of the challenges of gene therapy clinical studies and also of clinical studies in rare disease populations and things like that. And in our clinical trial, and when we did our informed consent, we did make a request for tissue at autopsy, but there is so much emotion involved. There are so many logistics involved, and it just did not pan out. And so we don't have a way of really determining which of these things was actually what led to this. So it is conjecture, right? It is based on information we have and other studies we've done. We're just making connections. And also a very important thing that should always be noted, especially for rare disease studies where our N is so small, there is oftentimes missingness of data because the kids are so unwell, they're not able to come back for studies and things like that. So that can cause you not to have sufficient follow-up, which again can cause some issues getting enough statistical data. But also it's just everything sort of is complicated. So much more. So as you're designing rare disease studies, how to follow up and get all of this information is what sort of becomes key. And also how these children are looked after by their families. If some children receive more care or more nursing care or more physical therapy or more whatever, those can also have an impact. So it's very hard to parse what is fully treatment related and what is actually also happening in the rest of their lives.
Right. That makes sense. And you're right. These are very small numbers. It is intriguing and perhaps it does sort of ring true, though, that the greater biodistribution is going to be better. I'm curious, in your preclinical study, did you see greater biodistribution in the neonate brain compared to the older brains? And did that also tend to correlate with the response you saw?
Yeah, we got great biodistribution there, but also, you have to know, like in neonate mice, there isn't the formed blood-brain barrier, and so essentially you get really good distribution. And we had so many behavioral parameters that we're assessing, and these were essentially performing like normal mice. So it was really good. But in the other thing, which I don't want to miss out on saying this, because this to me is even as important, is that 20 years after gene therapy, we also know that this did not cause any form of cancer. No hepatocarcinoma, no such things that have been of concern, long term and things like that. And I think this was extremely important to us also to point out that in addition to being, the longer survival, it was a longer survival with no malignancies.
Absolutely. Absolutely. And so now maybe again, this is such important work. I'm curious, maybe I can ask this question in two dimensions. I'm curious how we get more of this published. And so I guess one maybe from your perspective, is there anything you would have done differently in the clinical design or how you planned for long-term follow-up that you think would have made it easier for you to make this publication? And then conversely, what do you think the field should be thinking about as they try and implement something? Because I think I remember early days working in gene therapy at let's say a larger company that hadn't worked on gene therapy before. And people always saw the 5-year follow-up as that's regulatory mandated by the FDA as a little bit of a burden, and not the benefit that it really is, right? By the FDA sort of stating that at a minimum you have to do 5 years, that's actually a huge benefit because we do get that long-term data on these patients. But, I might argue it does, like what your data shows is that doesn't take us far enough, right? And so how do you think we can encourage more of this from our field?
So I think that it's very, what would we have done differently? I think that if we had, so, okay, we are very limited by budget, right? Everybody is very limited by budget. And so perhaps doing the full follow-up for a long period of time is not easy. But doing a subset of things does become, it is doable. And so so essentially, in our group, because we sort of run, as I said, like a small biotech with all of these different aspects, we have a group which we call CORA, which is Clinical Operations and Regulatory Affairs. And so we have a group of people who help us with all of this, who do patient follow-up and who do all of this. And a lot of this ends up being a phone call or an email, and you get that information. And, to be able to get access to patient data from the neurologist that they see and all of that, those connections that you make early on in your study with their pediatricians or their neurologists. And if that can be continued, I think that can help. We could have done better in that we could have put this in our IRB and all of that. We didn't have ability to do that detailed follow-up. We continued with our follow-up, but if we had been able to get access to their other data and have that information, that would have helped as well, without being so onerous on the cost issue, right? Because we would be getting data that was being collected irrespective. So that can help. And I think that if we had done that, that would have improved this, in that we may have had more knowledge on things like when did they get a GI tube, or when did this happen or that happen. That would have maybe flushed this out a bit better. And so I think, and how do we encourage other people to publish? I think what I would really love to see, and I don't really know how, is that we don't just publish our successes. Because here, I think we're very clearly pointing out our mistakes, not mistakes, but things we could have done better. And also the fact that it didn't work in some of the patients. It only worked in a subset. And that's not a bad thing. That's how we're going to learn how to do the next thing sort of better. And I think that as important as successes are things that didn't work, because then we don't have to repeat those same things again, and we just learn how to do it better. And I think gene therapy, to me, is an iterative process. And I think each clinical trial is going to inform the next and the next. And that's how we're going to be able to do it better.
Yeah, and you're right. It's really hard, especially when, you're starting out in that first study, right? Your phase one study, for better for worse, is going to be your longest term study. Like that data started earlier, so you will
Yeah.
have seen it for longer by the time you get to major regulatory successes later in a product's lifespan. One of the things I've seen, companies do at least is try to, if you will, intellectually kick the can a bit on what they need to do in terms of depth of follow-up by rolling over, patients in the phase one study into a long-term follow-up study that at least allows them to say, I don't know exactly what this needs to look like yet. I mean, they probably know what the first 5 years has to look like to satisfy FDA requirements. But then, if they are having success, or even if they're not, being able to say, I want to be able to simply add on an additional phone call once a year, as you're saying, to check in to see how things are going, for even if it's directly with the patient, for things that are event driven, right? Like, did you have a GI tube use begin? These kinds of things, it seems like it might not add up to all that much, but I think, looking at your data and hearing you talk about it, this could, thinking about more flexibly about what a long-term follow-up can look like, um, may greatly improve the data that we have as an industry to impact how we move forward in this field.
I totally agree. And also, I think the realization probably that no data is too little to
Mm,
share if you find the forum to share it, because I think it's really important and the next person is definitely going to benefit from it.
absolutely. Absolutely. All right. We've reached the end of our discussion. So I get to ask my very favorite question, which is, if you could wave a magic wand and fix one or more things, I'm not going to be particular about gene and cell therapy, what would you do?
It's as you said, I don't think I can say one thing, but I would love, especially for rare diseases, I really think gene therapy has so much promise, so much promise. And so if there were a way to do this thing that everybody talks about, is, have a platform kind of where a lot of this information is available, that you can kind of leapfrog over, some information to be able to do the next thing without having to do that first thing, I think we could get a lot more done. And so if there was such a magic repository of information, and that people were sharing, and being more available with their data, so that we can do more, essentially, I think that's what I would do. And then I really think that gene therapy has so much promise, but I do see that we are also getting to the stage that we are seeing some of the issues too. My dream is that people will give us the time to fix all the issues that there are, so that the magic can really be seen, because I think it has tremendous potential.
Absolutely. I mean, I think if you would have told me that you could basically, over double the lifespan, right, of a CLN2 patient with a therapy like this, if you told me that 20 years ago, I'm not sure I would necessarily have bought in. But this paper establishes that, right? For 3 patients, that's what you're seeing. And um, I think it suggests there is something there, that it's mediated by this therapy. And we just need to figure out how to make it more consistent across all the patients, and give everyone the best chance of success.
Yep, absolutely.
Dolan, thank you so much for joining today.
Thank you so much, Emily. Great chatting with you.